Welcome everyone to the recorded webinar series on LED-based photoacoustic imaging. My name is Mithun and I'm a Research and Business Development Manager at Cyberdyne. In this series, several key opinion leaders in the field of photoacoustics will talk about LED-based photoacoustic imaging and its preclinical and clinical applications. Today's very exciting talk will be about studies of precision and accuracy in LED-based photoacoustic imaging and will be delivered by Professor Sarah Bohndiek from the University of Cambridge. Professor Bohndiek completed her PhD in radiation physics at University College London in 2008 and then worked as a postdoctoral fellow in molecular imaging at both the University of Cambridge and Stanford University. Since 2013, Sarah has been a group leader at the University of Cambridge, where she is jointly appointed in the Department of Physics and the Cancer Research UK Cambridge Institute. The broad mission of Sarah's interdisciplinary team, the Vision Lab, is to develop and apply new imaging biomarkers that shed light on tumor evolution. They are also active in translating their findings into clinical trials. Sarah was recently awarded the CR UK Future Leaders in Cancer Research Prize and SPIE Early Career Achievement Award in recognition of her interdisciplinary research innovations. In addition to her research program, Sarah also takes an active role in teaching and mentoring the next generation of scientists. She has acted as an ambassador for public engagement and interdisciplinary research training throughout her career, and acknowledging her generosity in this regard, Sarah has received the MSCA Award for Nurturing Research Talent and has been invited to deliver numerous talks on career development at both national and international workshops. In this webinar, Sarah will talk about studies of precision and accuracy in LED-based photoacoustic imaging. With that, I will turn it over to Professor Sarah Bohndiek.
Thank you very much for joining today for my presentation on studies of precision and accuracy in LED-based photoacoustics. I'm Sarah Bohndiek and I'm Professor of Biomedical Physics at the University of Cambridge. I have to declare a conflict of interest in relation to the topic of this presentation, which is that I received research funding from Praxian Corporation, of which the Photoacoustic Imaging Division was later acquired by Cyberdyne Inc. First, I'd like to give you a short introduction to my team, the Vision Lab at the University of Cambridge. We develop and apply new multispectral imaging methods combined with advanced computational methods to study tumour evolution. Multispectral imaging itself refers to the capture of additional dimensions of light beyond the 2D or 3D spatial information that compose the image. Here we're collecting added dimensions of light colour, or wavelength, and also time, as we use our non-invasive methods to sample longitudinally, and that's how we look at tumour evolution. We use photoacoustics for deeper tissue imaging and endoscopic imaging, where we go directly to the site of interest, particularly in the gastrointestinal tract. To advance these methods, we use new developments in optics and photonics, as well as biophysical modelling and machine learning methods, which can help us to connect our recorded imaging signals to the underlying biology. We perform preclinical studies in mouse models, particularly cancer models, and also take these new technologies through to first-in-human clinical trials. In this particular talk, I'll focus on photoacoustics and give you some examples of some of the studies that we've done in preclinical mouse models. Why do we need new multispectral imaging tools to study tumour evolution? Well, our traditional imaging tools capture only part of the information that's needed to understand the evolution of a solid tumour. If we think about the tumour residing within the patient, I've got an example here of a breast tumour. The tumour is not a homogeneous mass. It's a very heterogeneous and complex dynamic organ in which we have a heterogeneous distribution of different cell types, also different extracellular matrix proteins that hold the scaffold of the tissue. We have lots of different chemical and physical signals that are playing into this tumour. So we no longer want to visualise the tumour as a mass. We want to look at the composition of the microenvironment of that tumour. If we think about our traditional radiological imaging approaches, such as computed tomography or magnetic resonance imaging, These are very good at giving us whole body scans, but they're relatively limited in their resolution with which we can study the tumour mass. If we go to the other end of the spectrum and think about microscopy methods that we might use for biological imaging, these provide us exquisite cellular resolution, but are often limited with resolution once we try to image larger fields of view or also greater depths. Photoacoustic imaging is a tool that has the potential to bridge these gaps because of the scalability with which you can operate photoacoustics. So photoacoustic imaging reveals the spatial distribution of optical absorption in tissue at high resolution. And this works by directing light at the tissue of interest. This is often pulsed light, and it's based on the absorption of that non-ionizing radiation in the tissue and the subsequent emission of ultrasound waves. So essentially we reconstruct an ultrasound image which contains biochemical information, and that's because light is absorbed by a range of different chromophores in tissue, predominantly in the case that we're interested in for tumour evolution, by hemoglobins in red blood cells in the chaotic tumour vasculature. Photoacoustics can therefore give us a very detailed picture of the tumour vasculature, and it can also be readily integrated into existing ultrasound systems, which means it has the potential for truly widespread clinical adoption. To give you an example of some of the sorts of imaging data you can get with photoacoustics when we're looking at imaging of tissue, blood concentration and oxygenation, we can think about implementing photoacoustics at the microscopy level. This is an optical resolution photoacoustic microscopy image of the vessels in a mouse ear. And here we're looking at the difference between arteries and veins by using the multispectral capability of photoacoustics, so measuring at multiple wavelengths and resolving the differences in absorption between oxygenated and deoxygenated blood. This is a similar example where there's a tumour embedded in the flank of a mouse who's breathing either carbogen or air, and you can see differences in the oxygenation of the hemoglobin in those tumour regions. Here's another example where now the tumour cells themselves are expressing tyrosinase, so we have a melanin pigmentation in each of the tumour cells, which gives us a distinct photoacoustic data compared to the vascular networks in which we have hemoglobin as the absorber. This is imaged with a Fabry-Perot Ettalon ultrasound detector, which allows us to build up quite a large volumetric imaging capability. We can also take data using a tomographic approach. This is again going back to more conventional ultrasound transducers, except now arranged in a cylindrical array around the mouse torso, where again now we have a subcutaneous tumour growing in the mouse, and we see a high level of deoxygenated blood correlating with the region of extensive tumour cell death as measured when the tumour is taken out of the body. And finally then we have an example from a patient study where now we've used a linear array transducer, and it's been coupled with a light-based photoacoustic excitation, and they've applied this to study breast tumours in a very large number of patients and derive different metrics that allow you to identify benign and malignant lesions. So there's a real breadth of information and length scales that are accessible with photoacoustics, but traditionally our photoacoustic instruments are relying on relatively slow and fragile as well as expensive nanosecond pulse lasers, and these result in bulky systems that need regular maintenance. There are also complex laser safety requirements for these systems for the operator, for the subject, and for the room in which the study is undertaken. In recent years, people have started to really explore LED-based excitation in more detail, and I'll just highlight a couple of examples here that got us really excited about the potential of using an LED-based approach. This is a paper from the Wang Lab at Michigan, where they looked at joints affected by inflammatory arthritis on the right, and normal joints arthritis on the left and normal joints on the right. And they've compared the groups quantitatively based on the photoacoustic signal and showed a significant difference between them. The Desjardins Lab at UCL investigated LED photoacoustics in the area of minimally invasive procedures, and here's an example of imaging in the human wrist, where they see strong photoacoustic signals in the superficial blood vessels that correspond well to the anechoic regions in the ultrasound. Here's a further example, now again in a preclinical mouse model from the Jokos Lab at UCSD, where they considered the feasibility of an LED-based approach for looking at stem cell imaging, here showing visualization of the DIR dye, which has been used as an effective contrast agent for cell tracking. And beyond these works using linear arrays, the Kothapalli Lab at Penn State used the combination of several LED sources in a computed tomography mode, showing visualization of data from a phantom at least at several centimeters of depth in orders of 100 micron resolution. So these studies encouraged us in my team to start to look at the potential of LED-based photoacoustics. And something that we always do when starting out with a new imaging modality is to consider how well we are able to quantify the imaging data from that system. And there are a couple of established roadmaps in this area, particularly one roadmap designed for imaging of cancer and a second roadmap designed for optical clinical implementation of optical imaging biomarkers. And these essentially follow three parallel tracks. One looking at technical validation, so looking at the precision and accuracy with which we can make measurements. The biological and clinical validation, which is in this case looking at how well the imaging signal connects with the underlying biology, and in addition, how well that can then be used clinically to make decisions. And finally, the cost effectiveness, so compared to the existing procedure that's done clinically, how cost effective is the new procedure. Now, photoacoustics, both with laser-based and LED-based system, I would argue, has been evaluated quite effectively in vitro, in animals, and in human initial trials. So we've sort of reached this first translational gap where we have to think about moving from those initial studies into using the imaging approach to measure parameters that can be used in a research setting. And this is a particular example from cancer research, but you could use this more broadly. So can we make measurements with photoacoustics that are reliable enough to test hypotheses in clinical cancer research in this particular case? To answer that sort of question, we have to start to evaluate in great detail the technical side of things in terms of precision and accuracy, and look at the precision with which we can measure the biological parameters. Then ultimately, of course, we want to move everything further over the second translational gap into application in healthcare systems. So in our case, we were able to do studies on an LED-based photoacoustic imaging array, and we used it for longitudinal preclinical imaging. And we evaluated the performance both for phantom imaging and for small animal imaging. So this is just a photo of the probe itself. This is the linear array ultrasound probe in the center, and these are the LED arrays on either side. And this is a dual wavelength LED array with excitation at 750 nanometers and 850 nanometers. For the studies in phantoms, we designed and constructed a 3D gantry, which would allow us to mount the LED-based photoacoustic array and then have a reproducible phantom positioning within a custom water bath. We then used that same gantry to place the probe on top of a custom water bath that was designed for coupling the ultrasound signals from a mouse, and that enabled us to do some in vivo imaging studies. Firstly then, to look at the studies in phantoms, we fabricated a phantom here from PVCP, and we included in it optical scattering and absorption, which allowed us then to identify different signals, photoacoustic signals in our LED-based photoacoustic image. There are different holes here to allow us to introduce different tubings at different time points, but here's an example where we've got tubes just at either end of the phantom here, and the signals that they generate in terms of photoacoustics. We use this phantom first for doing imaging over the time course of a normal imaging study. So in our case, we decided to do this over a few hours just to see how stable the output signal was. And here you can see the 750 nanometer LED data is always in red circles, and the 850 nanometer data is always in black squares. What was encouraging about both of the study looking at a single time course of 180 minutes, and also the later study, which we repeated the same experiment every day for the first week and then over the course of 30 days, was that there was basically no significant difference in the gradients over time. So these were not significantly non-zero, they were essentially flat gradients, and the extended study over 30 days resulted in coefficients of variation of only 7% for the 750 nanometer LED and 5.8% for the 850 nanometer LED. So these were quite good in terms of our ability to extract reliable data over an extended period of time. We also conducted a study just over one day by repeatedly imaging once an hour over 6 hours either leaving the phantom in place, which is the data at the top, or taking the phantom out and putting it back in again each time. And what you see here is essentially flat lines which indicate very low variation, and in fact if you calculate the coefficient of variation you're talking less than 2% in all cases. So for us these results indicated excellent temporal stability for longitudinal imaging. We then looked at whether frame averaging could enhance image quality in phantoms, trading off against the frame rate. One of the advantages of LED-based photoacoustics is that you can get very high repetition rates from the LEDs, which we can trade off if we want to enhance image quality by averaging these frames to enhance the signal-to-noise ratio. So here's the data from the 750 nanometer LED and again for the 850 nanometer LED at the bottom. And what you see is that in both cases, if you increase the number of averages, you eventually reach a threshold over which a maximum signal-to-noise ratio is achieved for imaging in the phantom. And in the case of the 750 nanometer LED, this happens after a somewhat larger number of averages as compared to the 850 nanometer LED. So if we went for the maximum amount of averaging or to obtain the maximum SNR, we'd be talking about frame rates of about 0.1 or 0.2 Hertz respectively if we were imaging at 10,000 or 20,000 averages. But we can easily, now we've done these studies, trade off and say, okay, what we'd like to do is get a 20 or 30 or 100-fold enhancement in SNR. So we're going to take a few thousand frame averages that will still be operating almost in real time, or in some cases actually in real time. But by doing the averaging, we're giving ourselves an enhancement, a little boost in the signal-to-noise ratio that's available. We then went on to do some studies in vivo, and you can see the videos playing in the background here as the mouse breathes medical air or 100% oxygen. So this hemoglobin oxygenation modulation is something that's well established now in the field, using a breathing gas challenge where the mouse is anaesthetized under those two different circumstances, either breathing medical air or oxygen. And here are some examples of some still images zoomed in here where you can see the oxygenation that we measure from the region of the liver in the mouse. This is done using the dual wavelength imaging, so we take both 750 nanometer and 850 nanometer data and extract a relative measure of oxygenation based on the ratio of those two. The coefficient of variation over time for multiple cycles of changing the gas breathing was found to be less than 10%. So we extract the delta XO2 here for each of the up and the down cycles, and we then look at the variation from a single mouse first, and we see similar changes in SO2 from air to oxygen and from oxygen to air. The coefficient of variation of the overall oxygenation when the mouse is breathing air is about 3%, and similar when the mouse is breathing oxygen. We then studied a second mouse to see how the biological variation compared between those, and as we expected, we would get an enhancement in the coefficient, an increase in the coefficient of variation, but still within the region that you might expect for a biological study, and certainly within the values that we've seen before. We next went on to look at the kinetics of Indesign in green contrast agent administration, which can also be visualized and quantified. So again, this is on the liver region. We've now got the baseline signal pre-injection and the signal post-injection of 48 nanomoles of Indesign in green, imaging undertaken at 750 nanometers. And now again, we normalize in this case the mean pixel intensity to 1 at its maximum, and we show you the kinetic curve, and the temporal variation in the uptake compares favorably to liver kinetics we've observed previously in other systems. So this is just a hint that we can also then start to look at contrast agent administration with this system, but there are many more studies that would still need to be done in the context of both technical and biological validation from our side, and I'll talk a bit more about that on the next slide. So to summarize, LED-based photoacoustics can provide a cost-effective alternative to laser-based excitation, allowing the potential for photoacoustic imaging that I illustrated in terms of the multi-scale imaging approach to be applied in a more cost-effective manner. And particularly thinking about this from a translational perspective, having a cost-effective excitation source might allow us to apply photoacoustic imaging in a wider range of indications. In our case, we looked at the application of LED-based photoacoustics in stable phantoms, showing excellent longitudinal reproducibility over minutes, hours, and days. We also looked at visualization of the mouse liver in vivo as a kind of first step towards biological validation by applying oxygen-enhanced imaging, cycling between two breathing gases, and also intravenous administration of a contrast agent, and showing that these could be detected and reliably quantified with the dual wavelength LED-based system. But there are lots of future studies still to do, as I mentioned. First, these were all done with the probe mounted in a gantry, so it minimised the effect of the operator, and what we'd like to do is look at the impact of handheld operation on the reported coefficients of variation. We'd also like to investigate the potential for applying further data analysis and processing. So one of the challenges I mentioned earlier is that even with multispectral photoacoustics, the absolute quantification of tissue absorption is challenging. We'd like to look at whether we can apply fluence corrections to improve quantification of the biomarkers that we've looked at here. I'd also like to highlight at this point that if you're interested in studies of precision and accuracy and general validation of photoacoustic imaging, there is an international photoacoustic standardization consortium that I chair. We now have over 125 members, and we have 3 themes that are operational, looking at both development of new phantoms, data acquisition and management, and designing multi-center studies to move photoacoustics from that initial single-center research phase into a larger and broader effort, which you can find out more about on our website, which is ipask.science. With that then, I'd like to thank the members of my team who were involved in this study, particularly James Joseph, who essentially drove all of the LED-based photoacoustic work in the lab, our collaborators for undertaking the study, and thank you for joining to listen to the presentation. If you do have any questions, then please feel free to email me and I'll be happy to try and answer them. Thank you very much for your attention.